Hello everyone. Let's cover the last segment of this chapter. In the last segment of this chapter, we are going to look into viruses and we are going to particularly address the flu virus. And again, I'm going to take the time to break things down. If there are things you don't understand, please let me know and bring you questions on Tuesday when you come to class on Tuesday. All right. So typically, the flu is a disease of the upper respiratory tract and unfortunately can also expand in the lower respiratory tract. Now, when it expands in the lower respiratory tract, that is pretty much a bad news, even though most of the time it's not going to be fatal. Now, know that the flu is caused by a virus called influenza. We are going to take a look at the characteristics of influenza. It is estimated that about 20% of the population, the human population, is affected with influenza. Now, you know that the flu, it's a seasonal infection. It's an acute infection that goes as fast as it comes. When we consider the different types of influenza that are out there are really three major virus types. These three major virus types fall into type A, that actually causes very serious diseases that can actually lead to death. We have type B, that is less serious, less severe, nevertheless, it causes discomfort. And we have type C, that is minor, doesn't cause too much issues. Now, what are the symptoms and the signs of infection to influenza? Well, typically we're going to have a fever, headache, sore throat. I kind of say things backwards. I said fever, but I underline headaches, but you understand headaches, fevers, sore throat, muscle pain, and sometimes it's actually the entire body that is going to be painful. And that happens relatively fast within a few hours to about a day when you start really feeling weary and tired. The symptoms can further be exacerbated with the development of dry cough, right? Uh, and and also the situation can worsen over time. Now, flu lasts about few days, most of the time about a week, not much more than a week, even though cough can linger, fatigue can linger, weaknesses of the muscles can actually linger for over a week, even if not couple of weeks. Occasionally, you can have a swelling of the brain and the liver can be damaged. This is actually known as the Reyes syndrome, right? And the flu virus is not the only virus that actually triggers this Reyes syndrome. Now, the infectious agent that causes flu, influenza, it's actually a virus that is an enveloped virus. One. Two, that enveloped virus is going to contain RNA, is going to contain RNA as its genetic information. Now, along with the envelope, we are going to have a number of proteins that are embedded. Typically, two kinds of proteins. I want you to remember that. We have in the envelope the glycoproteins, okay, that constitute the spikes of the envelope. And these spikes occur in two kinds. One is called hemagglutinin, okay, hemagglutinin. And another one is called neuraminidase, okay, two of them, hemagglutinin and neuraminidase. Now, here we have the hemagglutinin. Here we have the neuraminidase. So, these molecules are the one that are characteristic on the surface of Haemophilus of influenza. All right. So, in terms of genetic information, the flu virus is going to have many fragments of RNA. Okay, 8 of them. These 8 fragments of RNA actually go alongside with a small protein that is called a nucleoprotein, right? Also, I'm not sure why this is not represented here, but the influenza virus comes also with an enzyme that is called a reverse transcriptase. I want you to remember that enzyme, reverse transcriptase. Let me write that. Reverse transcriptase. Now, just so you know, when we consider a cell has DNA as the genetic information. That genetic information is expressed by going through a process called transcription that involves an enzyme called RNA polymerase. RNA polymerase converts DNA into RNA, and then the ribosomes are going to convert RNA into proteins. The reason why I explain this, it's because the influenza virus that has an enzyme called reverse transcriptase, it's actually going to do the opposite job that happens between DNA and RNA. In other words, because the genetic information in this virus is RNA, when the virus comes in, RNA is first of all converted into a DNA molecule, a single-stranded DNA molecule, right? And the reverse transcriptase is the enzyme that does that job, okay? It converts RNA into DNA. And the reason why this happens is very strategic. I will explain a little later, but let me close that parenthesis. So, we have 2 spike molecules, 2 proteins, hemagglutinin and neuraminidase. And so, hemagglutinin stands for HA, or HA stands for hemagglutinin, while neuraminidase is represented by Na. Okay? So, HA is essential for the attachment of the virus influenza on epithelial cells. Now, for the attachment to happen, there is a need to a specific recognition between the signal molecule and the receptor that attaches that signaling molecule. I want you to remember that. And then we have the Na protein. That Na protein is critical for the release of the molecule, for the release of the virus content inside of our cells. Now, let me close that parenthesis. I want you to remember two spikes, the hemagglutinin and the neuraminidase, right? Both are antigens because they are very immunogenic. All right. Having said that, over time, as we improved the molecular tools for diagnosis, we noticed that, well, there were many spikes of different strains of influenza. Specifically, in 1997, there was identification of an avian flu strain that was called H5N1. That H5N1 avian flu strain caused the death of many birds. Then another outbreak I want to mention, it's the one that happens in 2009 that came from swine, the pork, right? The pigs, right? Swine flu. That strain was H1N1. Now, H1, it's basically a number that is given to hemagglutinin, and N1 is a number that is given to neuraminidase antigen, right? So, also know that there are different versions of H and different version of N. In fact, there are 16 versions of H that are known and 9 versions of NA that are known that are part of the subtype H1, right? H2, H3, and N1, right? That actually are able to infect people. Now, let me digress. Now, we say that in 1997, we talked about the outbreak of avian flu, H1N1. And then in 2009, we had an outbreak of swine flu, H1N1. But now we are talking about strains that are able to infect people. One must wonder, wait, Dr. Jean, yes, obviously, we do have strains of influenza that typically infect humans. But when it comes to avian strain and swine strain, how? How does it jump from swine to human, from avian to human? What is going on there? We are going to address that. So before we address that, know that the virus can be spread by air, right? It's an airborne disease. I mean, you know this, so this is not new to you. We take that in by inhalation, and the viral particles are within droplets, right? When we sneeze, and especially if we sneeze in our hands, we're going to shake people's hand, or we can breathe the sneeze of somebody who is sneezing by us, things like that. Now, know that a virus is called virion when it is outside of the host. Let me repeat that. A virus is not called a virus when it's outside of the host. It's called a virion, right? We are going to call a virion a virus when that virion attaches to the target cell and gets internalized. At this point, we are talking about a virus. So we don't use the term virus until the virion is inside of the host. Okay? I want you to remember that. So, as the virus gets in the host by way of attachment, right? This virus is going to release its content inside of the host. But what is the content? The contents, it's basically RNA. That RNA with the reverse transcriptase is going to be converted from RNA to DNA molecule, single-stranded DNA molecule. The reverse transcriptase does that. Now, as now we have in the cytosol a one single strand of DNA, the cell, our cell, is going to wonder, wait a minute, so this is a piece of DNA. A piece of DNA is not supposed to be, first, a single-stranded molecule. That's one. Two, in the cytoplasm. That's the second thing. So our cells are going to see that as, we need to do something here. The something that is going to be done, it's that the single-stranded DNA molecule, right, that is a reverse copy of the RNA viral particle, right, is going to go through the copying of the second strand of DNA, so that now we are going from a single-stranded DNA molecule to a double-strand DNA molecule, which is the normal form that our system recognizes that, oh, this is normal DNA. And once that RNA is converted into DNA, that is now a double-stranded DNA molecule, then the cell is going to think that, you don't belong in the cytoplasm, you belong in the nucleus. So that DNA is going to be carried from the cytoplasm to the nucleus. And eventually, that DNA is going to insert in our chromosome, be expressed, and this is how, over time, you are going to see the expression of the viral particles, and specifically the viral proteins, right? And the virus can reconstitute that way using our machinery, right? So, so that's a cycle that is very important to understand. And once our cells produce viral particles, eventually there is going to be assembly of the virus, and the viral particles are going to be so numerous that when they leave, they're going to cause our own cells to burst. And this is where things become nasty for us, right? Fortunately, this is this is an acute infection that is actually going by relatively fast, and most of the time, the flu episodes are mild episodes, right? Because the one that can really hurt you seriously, even leading to Reyes syndrome, are not very common. All right. So, what else should I tell you here? Okay, so what we notice is that when we consider a particular season and we consider the flu happening in that season, and we start tracking the strains of influenza during that season, we notice that there are many versions of a particular strain. The question we may ask is, what are the consequences of these many versions? Well, the consequences, it's that we are going to see what is called antigen drift. Now, what is antigen drift? Let me digress. I told you that the virus comes with an enzyme that is called reverse transcriptase. that reverse transcriptase job is to convert RNA into a complementary strand of DNA, one single strand. I have to say that what we notice is that that enzyme, the reverse transcriptase, does not have a proof reading activity. In other words, when the enzyme is doing its job, it can make mistakes. And in fact, it does make mistakes. The problem, it's not that it makes mistakes. The problem is that it does not know how to correct the mistakes. So that when we get infected with one strain, by the time we spread the virus around, we are spreading many subversions of that strain, so that we are going to have multiple form of the strain. This multiple form of the strain over time can become different one from another. And this is what we call antigenic drift. And now, to be specific again, antigenic drift, we are going to specifically look at what is happening to the neuraminidase antigen and to the hemagglutinin, right? We're going to notice that these antigens actually are changing slightly from one version to another, so that two people can be infected with two different versions, three different versions that are not 100% alike, even though more than 90% or 99% even, they are going to be alike. The differences are going to be enough for these two strains or substrains to actually exhibit kind of different symptoms, right? I want you to remember that. And this is actually the reason why we have to develop new vaccines every season, because there is a genetic drift, right, of this strains. Okay. Now, on the other hand, we have another concept that is called the antigenic shift. That antigenic shift is caused by genetic shift. That genetic shift, it's simply that a swine flu that is actually able typically to recognize swine, okay, pigs, right, hogs, right? That strain can actually experience some genetic changes that are drastic enough to actually go from not just recognizing target cells in the swine, but can also recognize target cells in humans. And this is because of the proximity, because we have farms, right? Say we have this swine in our farm, and we come in contact all the time with these animals. There is a strong probability that some versions of the swine virus can actually become infectious to human. By the same token, the flu, the avian version, can also experience a genetic drift, a genetic shift, right? That is going to be such that the spikes that were not able to attach to our receptors, but only able to attach to birds, is now going to be able to attach to humans. So we're going from one species of animal to another species of animal. This is a complete shift, right? That's why we call it antigenic shift or genetic shift. So we talk about antigenic shift when we go from one strain able to recognize one particular target cell in a species to being able to recognize a target cell in another species that is completely different, right? And that we do see this because of the proximity, right? That exists between these animals and us. So remember, the antigenic drift and the antigenic shift, it's not the same thing. So now, when we track a particular infectious particle, say we have this guy right here infected with this virus with a particular type of H spikes. Now, this H spikes, when the reverse transcriptase is converting RNA into DNA, can make mistakes. And these mistakes can happen within the H antigen. When that happens and that the virus multiply, they are going to express on the surface modified versions of the H antigen, right? And so this is during mutation one, after mutation one, right? And now, in this individual now, we can see another change still on the H antigen, so that we have not one single change anymore, but two changes. So as we go from this guy to this lady right here, we have different versions of the H molecule. Now, understand that what happens with H can also happen with N. I want you to remember that, okay? So that you can have many combinations of strains or subtype of strains that are going to be different one from another, right? So that what this guy here is giving this lady is going to be different from what this lady is going to give this lady right here, okay? So this is within the same group of organisms. When we see this change, this is what we call antigenic drift, okay? And then we have antigenic shift. In antigenic shift, we have one version here able to be, that is specific to a bird, another version of the influenza here that is specific to human. Modification can be such that these versions, okay, can actually be able to infect a pig. As a matter of fact, what we notice is that in terms of research, we see that, well, the bird flu and the human flu virus kind of mingled somehow their genetic information, so that we have a mixed version right here in the swine that is able to infect the swine. By the same token, right, this swine now can pass the version to humans, and well, that's not a good news. I mean, either way, whether we are passing our germs, or in this case influenza, to the pig, or the pig is passing us their influenza, that is not a good news, right? When it crosses species, it's not a good news. And understand that because of our close proximity with the pets that we are transferring a lot of stuff to pet. It's not just the pets that are transferring germs to us. I want you to remember that. Now, here we are going to track a little bit the spread of the H5N1 avian virus. Now, we can see that that we have areas that are reporting the virus in poultry. Now, look at how it is spread across the world. Okay? Spread across the world. And then areas occurring where the virus is occurring only in birds. Take a look at the different areas, right? So there was a spread, okay? There was a spread from birds, the wild organism, to poultry, the domesticated organisms, right? Which eventually landed in humans. Okay, so this slide is a summary slide as we've been saying. So please go through the slide. I'm not going to go through it again because I already explained a whole bunch of things. So this is going to conclude this chapter, and this is where we are going to actually resume we're going to start a new chapter when we meet on Tuesday. Until then, take care of yourself. Bye now.